First, welcome to First Parish Unitarian Universalist Church. We're non-denominational. We steal the best from many different faiths, and we are a leader in liberal causes and justice. With that, without further ado, I'd like to introduce Sean Ryder, and he'll talk to us about mRNA and his book and all sorts of very interesting and important topics. Thank you.
I need to be here by the microphone so you can hear me better. Okay, good. Bottom line, very nice of you guys to come out today to listen to me, and I'm glad you could be here. I want today to be more of a conversation than a talking head presentation on the screen. So I invite you to interrupt me, to ask me questions, to challenge me to explain myself if I'm ever using words or terms or concepts that are totally alien, because certainly what I learned in high school and college for molecular biology is very old news. What my children have learned is far more advanced than anything I've ever learned. And so I'd like us to all be on the same page by the end of today, if at all possible.
If you ask questions, Sean, would you mind repeating the questions to the microphone so that it can be picked up?
You got it. I will repeat questions into the microphone. By way of introduction, my name is Sean Ryder. I'm a professor at UMass Chan Medical School. I study RNA biology, and I've been studying RNA biology since about 1996 when I started a PhD program at Yale University. Before that, I was an undergraduate student at the University of New Hampshire, where I got a bachelor's degree in biochemistry. I've been grateful and glad to be involved in academic research for the past 30 years now. When I think about it, it's been a true blessing. You can learn more about me on my website, everydayrna.com. I don't really want you to read this. What I want you to know about me is that I'm an average everyday person. I'm a normal guy. I change my own oil. I do my own car repairs. I mow my own grass. I cook my own dinner. I raise my own kids. I'm just a normal guy just like you. Okay? None of this really matters. The only thing that I want you to pay attention to is right down here at the bottom. In order for us to have a meaningful conversation and in order to build trust, I don't invest in any biotechnology or pharmaceutical company in any way, shape, or form. In fact, all of my investments, both in terms of my children's education accounts and my retirement accounts, are in broad index funds, ETFs, or mutual funds. I really honestly don't know what they're invested in, and I'd like to keep it that way. And so I don't stand to profit in any meaningful way from the information I'm about to share, other than I did write this book, which I'm very proud of and just came out in October. So it's brand new to the shelves. You can find it on Amazon, but they take about a month and a half to deliver for reasons I don't understand. I do have a box full of them with me if you're interested in it. I do make a small royalty every time I sell a copy, so I do profit off of that. Sorry. My goals for today's presentation are for us to all get on the same page about what a vaccine is and what they do. I'd like for us to talk about why some vaccines work really, really well and why some vaccines we seem to have to take over and over and over and over again throughout the course of our life in order to maintain some kind of immunity. I'd like to talk about the different types of vaccine technology and how they've changed over the past 150, 200 years of vaccine research. Most importantly, I'd like to talk about what I'm expert in, which is mRNA, how it's being used as a vaccine, and what the issues are with that, and what the promises are with that. And last but not least, I'd like to talk a little bit about how vaccines are made, so you get an understanding of what's in a vaccine and what's not. Okay. So I promise I won't show very many medical pictures today, but I have to show this one because many people in the room will not know what smallpox is. And that's because smallpox is an infectious disease that's been almost completely eradicated. As far as I know, there's 2 vials of smallpox left in the world, one at the CDC in Atlanta and another in Moscow, and that's it. Smallpox had a 30% death rate. So if you want to talk about case fatality rate, 3 out of 10 people who caught smallpox would die from it. Those who survived were often disfigured. So we have 2 children here. This is a photo that was taken in 1901. One had received a vaccination against smallpox, the other had not. You can guess which one did not. Those who survived were often disfigured. You may have heard the phrase pockmarked face. That's from scarification from smallpox infection. This is a disease that was endemic in the human population. We have evidence of smallpox infection dating all the way back to 3000 BC. You can see pockmarks on the face of Egyptian mummies. And it's thought that Ramses II died from a smallpox infection. The last known case in North America, I wrote 1952, but I'm pretty sure it's 1949 in Texas. And then the last known case in the world was in Somalia in 1977. Okay? Vaccines were released, that's not a great way to describe this one. The discovery of the vaccine that's been used almost continually since it was first discovered to about 1980 has been super effective to the point where smallpox is no longer a global threat. And anybody who was born after 1980 almost certainly did not receive a smallpox vaccine. People who receive smallpox vaccines these days are people who travel the world into areas where other pox-like diseases are endemic or sporadic, or not pandemic, but local outbreaks, things like mpox or monkeypox. So this is how vaccines started, trying to get around this disease. And the story begins in the late 1600s. And so the woman here is Lady Mary Wortley Montagu. She was an English aristocrat, and she was married to Lord Montagu, and together they spent time in the Turkish Ottoman court. Now, her brother died as a young boy from a smallpox infection. And she also had smallpox, but she survived and she was disfigured from the disease. None of the paintings show that, but that is the truth of it. And so when she was in Turkey, in Constantinople, in the Ottoman court, she learned of a practice called variolation. And what this practice is they would take a needle or a lancet, and they'd go to a person who had a smallpox infection, and they'd dip that lancet into one of the pox, one of the pustules that would form, and then they would take that fluid and they'd stick it under the skin of somebody who'd never been infected with smallpox. It's kind of like having a chicken pox party. Does anybody remember those? That's how I got chicken pox. The idea was, if you're exposed to a little bit of the virus, maybe you won't get the disease, but maybe you'll become immune to the virus in the future, right? And so that's the very basic beginnings of vaccines. It wasn't called vaccines, it was called variolation. And the idea is small dose exposure to a limited amount of vaccine might not cause disease, but it might confer some kind of protection. And unfortunately, about 1% to 3% of people who were variolated in this way in England died from smallpox. So the side effects of variolation were not very good. It's also known that other blood-borne diseases were spread in this way. So syphilis was very prominent in England around this time. And syphilis was spread from smallpox-infected persons to children through variolation. And so that was state-of-the-art late 1600s. The next big advance, and really the dawn of vaccine research, came in the mid-1700s, mid to late 1700s, And the person responsible for bringing this to our attention is Sir Edward Jenner, who was a medical doctor working in London. And so he had heard rumors that milkmaids in the north didn't catch smallpox. And so you may have heard this story before, but these milkmaids were exposed to a very similar disease of cows called cowpox. And so cows would have pustules on their udders. It was caused by a very similar virus. They didn't know that it was caused by a virus or anything at the time. They just knew that the pox looked similar. And these milkmaids would milk these cows, and they'd end up getting what we call zoonotic transmission. They'd catch cowpox from the cow, and they'd end up with pustules like this on their hands, which looked a little bit different from smallpox pustules. They were bigger. Okay? So this is what a cowpox would look like on the cow udders. This is what the milkmaids would get. When the milkmaids recovered from cowpox, which did not cause serious disease, did not spread all over the body, did not kill 30% of the people that were infected, when they recovered, turns out they never caught smallpox for the rest of their life. So somehow, inoculation with this similar disease of an animal was able to confer protection against smallpox. And so what Ed Jenner did was he popularized this idea. He turned it into a practice. And the practice is what we now call vaccination. He called it variolae vaccinae. Vacca being Latin for cow. What it literally means is variolation from the cow. Okay? So this is what, whenever somebody asks you what vaccination is, that's what it is. That's how it started. And so, this also birthed the dawn of the anti-vaxxer movement. We tend to think of this as a current phenomenon or something that's come about globally over the last 10 to 20 years. But in truth, anti-vaxxers have been around since vaccines have been around. The idea that you could take some fluid from a cow and put it into a human was something that was abhorrent to many people living in England at the time, including many medical doctors. And this political cartoon from James Gilray, who's called the father of the political cartoon, showcases what they thought might happen, with little cows erupting out of the faces of people who'd been inoculated, out of the mouths. This person's growing horns. And so the idea is that, are we going to turn into cows? This was a legitimate fear. There's books written about the symptoms of people thinking they're turning into cows, children growing cow hair, little horns starting to grow out of their head. None of that actually happened. And when it became obvious that people weren't literally turning into cows, the fear turned into, well, are they being made stupid, like cows are stupid? Hate to put it that way, but that was the idea. So anti-vaxxer is nothing new. But the truth is, this worked. It worked a lot better. Less people got sick, less people died. Cows didn't have syphilis, so syphilis wasn't transferred from cow to human. And over the course of 100 years plus, this technology wiped out the disease. Now, nowadays, the vaccine isn't made in cows, it's made in cell culture, but the idea is very similar. So if you ever get an Mpox vaccine, this is where it came from. Okay. The other, and the second major advance in vaccine technology came from a disease called yellow fever. Now, yellow fever was really nasty. It was a hemorrhagic fever. So if you've ever heard of Ebola or Marburg or one of those types of viruses, the people who would contract yellow fever would die in a horrible way, really, with bleeding caused by very high fevers, very high temperatures, internal organ failure. And it was a scary disease. And it was found mostly in South America, but there was also a disease that was very similar in Africa. And researchers didn't know if they were the same disease or not. And so they'd travel from Africa to South America to see if they could collect samples of this diseased material, figure out what was causing it. Many of those researchers died, right? Most of them caught yellow fever and died. Fatality rates were very similar to smallpox, about 25%. We still have yellow fever today. It's not a disease that's been eradicated. But we now have an effective vaccine. It was released in 1937. But cows don't get yellow fever, right? So how did we get this vaccine? So it turns out one of the things that the researchers were trying to do when they were collecting this virus material from patients is figure out what other animals might be able to contract this disease. African monkeys seem to be immune somehow. But Indian macaques were susceptible. So now we had a way to grow the virus, and by we, I mean researchers in the 20s, so 100 years ago. We had a way to grow the virus. We just needed these macaques from India, okay? And over the course of many passages through macaques and continued studies, it was found that a variant of the virus emerged that could infect the brains of mice. And after over 100 passages of the virus through mice, they would then infect macaques. And now the macaques barely developed any kind of disease. But when they were presented with a dangerous form of the virus, they were completely protected. And so this strain is called strain 17D, and it is the first example of a live attenuated vaccine. Has anybody heard that term before? Live attenuated vaccine? If you ever get the flu shot nasally, that's a live attenuated vaccine. So this is a live virus. It's just been weakened dramatically by passaging through other animals for now over almost 100 years. It still confers strong immunity, but it's not dangerous. Now, polio is probably the beginning of the modern vaccine era. This started, the vaccine research had a big jump in the 1950s. A gentleman named Jonas Salk. And what we know about polio is that most cases were asymptomatic. A lot like COVID, right? There's a number of cases that were asymptomatic. With polio, 95% didn't even know they had it. But about 1 in 200 or so, the infection would move into the nervous system, and they'd have what's called poliomyelitis. And that was a disease that was really devastating. So about 5 to 10% of those people ended up losing the ability to breathe on their own and would end up in an iron lung. Many people damaged their nerve cells so badly that they had permanent lifetime paralysis, including this boy who's using canes and braces to walk, including former presidents of the United States, FDR. So now we have a vaccine, and I don't know anybody that's had poliovirus, except for one person that we met at the protest on Saturday last week, if you happen to be there. So what's interesting about the poliovirus and what worked for Jonas Salk was he found a way to grow the virus, and in this case he used livers from monkeys, I believe. He was able to grow the virus, but then he was able to chemically kill it so that it could not possibly cause infection. Okay? He found the right mixture of chemicals. It's mostly formaldehyde, as it turns out. Any of you guys dissect frogs or anything like that? That horrible, awful smell, that's formaldehyde. And so he was using formaldehyde to kill these viruses, and then he would use that to inoculate people. And then that inoculation would confer them resistance to poliovirus. Okay? And so doesn't always work. They tried the same thing with the yellow fever vaccine, but the chemicals prevented it from working as a vaccine. So these things have to be tested and developed individually. But this was the first example of a killed vaccine or a dead vaccine virus. Most of our influenza vaccines that we receive use this technology. I want to do a little bit of an aside before we move on. And tell me if you can't hear me. But as an aside, the experiment that Dr. Jenner did to show that cowpox vaccine conferred resistance to smallpox. Can anybody guess how he might have tested that? No. Yeah. So the way the experiment was done, first it was done on a volunteer boy. And the way it was done is he was vaccinated with cowpox. And then he was inoculated with smallpox, right? So he was given an infectious agent that, should he not have been protected, had a decent chance of killing him. But it didn't. So then they moved on to prisoners. And they had a very hard time recruiting volunteers because of the whole cows are going to grow out of your face kind of thing. And that was true before as well with variolation when that first started in England. So we do medical research very differently now. I want to make that clear. But in the old days, what people would do is they would treat you with a vaccine and then treat you with the virus. And you didn't necessarily have to be a volunteer in order to get this treatment. And that's not okay. All right. So lessons from the past. Vaccines provide protection from infection without serious disease. That's literally all they are. Infection with one virus can sometimes lead to protection from another. The only reason cowpox gives protection from smallpox is that the two viruses are very similar. Okay? And they mutate very slowly. Passaging viruses through a non-native species, if you can find one, can weaken it so that it doesn't infect us as badly, so that we don't get a serious disease. That takes decades, tens of years, but it works. In some cases, vaccines can be killed with chemicals, and then if you can grow enough virus, kill it, you have an effective vaccine. Doesn't work for every virus. There's no HIV vaccine. There's no vaccine against rhinoviruses, which cause common colds. Doesn't always work. And most importantly, modern medical practice does not allow for human testing without informed consent, nor does it allow for risking people. Yes?
In all these cases in history, they didn't really know what they were doing, did they?
No idea.
It was experimentation.
Right. Humans were experimental subjects, in some cases unwilling experimental subjects. And that was true for hundreds to thousands of years. Yeah, it's gruesome to think about. Okay. So let's talk about viruses, what they are, right? So we've all heard of viruses, right? We've all heard about COVID. We grew up with colds and influenza and all kinds of different diseases. We know about HIV. We know about hepatitis. These are all different viruses. But what is a virus, really? Is it a living organism like a bacteria? No, not really. It can't replicate on its own outside of a host cell. It's a parasite. And all it really is a little package of some form of nucleic acid, often DNA, sometimes RNA. It's surrounded by a protein shell, and in many cases, that protein shell is surrounded by another layer of what's called a lipid bilayer. Now, lipid bilayers are what surround all of our cells. Okay? Our bodies are made up of millions of cells that are separated from each other by structures that look very similar to this. They're called lipids. And then embedded into that lipid membrane, we call it a membrane, are proteins that target it to the right host cell, so it knows which cell to attack. It's just like a little packet of nasty. That's what a virus is. Okay? And so what do they do, right? What does this little thing do? Well, it hijacks your cells. Okay? So that little targeting protein binds to proteins that are present on the surface of a cell. Some cells, not all cells. This is just, we'll call this a blue cell. Who knows what it's involved in? The virus binds to the right cell type. It gets incorporated inside the cell, and then it releases its genome, DNA or RNA. Let's say it's RNA in this case. It engages with this little orange thing, which we call the ribosome. I'll tell you about that in a minute. And that makes proteins that the virus is encoded in that RNA sequence. Those proteins are necessary to assemble new viruses. Okay? So you're hijacking the cellular machinery to make your own stuff. It's also hijacking the cellular machinery to replicate its DNA or RNA genome. And then these come together to form new infectious particles, which then either break out of the cell, killing the cell, or emerge from the cell through a budding process. Basically, the cell can't do its job the whole time it's infected because it's doing virus work. It's like if you get a virus in your computer. That's why they call them computer viruses. You can't open your email because it's hijacking your subroutines. Same deal here. You can't make your proteins because the virus is making its proteins. Make sense? That's what a virus is. All the viruses are different. They encode different proteins, they encode different genes. They do this hijacking in different ways, right? Like COVID and influenza affect our upper respiratory system. HIV infects our immune system. Hepatitis affects our liver, right? So every virus seems to be targeted to different cell types. And that's exactly what that external protein is. It's a targeting protein. And whether it infects a cow or a human or a sheep or a dog, or birds and humans, depends on whether we have similar proteins on our cells to those other species, right? So birds get bird flu. We also get bird flu. It's because the protein receptor that the flu recognizes is very similar in us as it is in birds, at least for some subtypes of the flu. That's what's called a reservoir species. So the flu is out there in birds. And sometimes bird flu passes into humans. And when it starts spreading from human to human, it becomes a problem. Okay. And they probably see it the other way. All these humans carrying flu around, spreading it human to human. Every so often, it transitions into birds. So what do our cells do? Like they're not just going to take it quiet, right? They're going to mount some kind of defense. And this defense is going to take care of this virus so we can get healthy again, so we can go back to work, so we can go do the things that we love to do. And this is a complicated slide, and I don't want you to memorize any of this stuff because it's not super important. But I put it up here because what I want you to know is that the immune system is complicated, right? You may hear that your immune system makes antibodies, and antibodies inactivate viruses. Both of those things are true, but your immune system does way more than that, okay? And the strength of the immune response matters. The types of cells that get infected matters in terms of how strong the immune response is. How sick you get matters in terms of how strong the immune response is, okay? So we have a virus here entering a cell. This cell happens to be a macrophage. It's a cell that's specialized in eating things that aren't human. So if you have non-human stuff in your bloodstream, the macrophage detects that as bad and says chomp. It doesn't literally say chomp, but it engulfs that particle, that virus, that whatever, and then it destroys it. But it does more. What it does is then it starts to present parts of that virus on the surface, right? As a warning. to other viruses. No, not as a warning to other viruses. But because what that then does is it activates the T cells and B cells, other components of our immune system, to do their job. Okay? Some of the T cells regulate how strong the immune response is. Some of the T cells are called killer cells, and they secrete all kinds of things that are bad for viruses and things that are infecting you. These T cells also activate B cells, which produce the antibodies that we talked about. And they also produce memory cells, which allow the body to mount a more rapid response the next time it sees the same virus. And it's how strong this memory response is and how long it lasts that defines how good your vaccine is, really. So when I was a kid, I had a measles, mumps, rubella vaccine. I probably had a couple, 3 doses of it. But I've never had it again, nor is it recommended that I get it. My doctor wants me to get a COVID vaccine every 2 years, an influenza vaccine every year, and a pneumonia vaccine every year. That's because these types of infections and these types of vaccines don't mount as strong of an immune response. And that's why you need to keep retraining your immune system. There's another reason, and that's because influenza, COVID, other types of viruses like that mutate a whole heck of a lot faster than other viruses. So they evade this memory by changing a little bit, just enough to get around your memory of past infection. Am I making sense so far? Stop me if I'm talking complete, utter nonsense. Okay. So, I already told you about this. Vaccines stimulate a strong immune response. Our first 3 vaccines that we talked about, the smallpox vaccine is a similar virus. It's cowpox vaccine. Now it's called vaccinia. Again, vaca meaning of the cow. People probably like that a little bit better. Yellow fever vaccine is a weakened actual yellow fever vaccine that doesn't cause strong disease. We've had that since 1937. The polio vaccine is chemically inactivated. We've had that since 1955. Those 2 vaccines are still in use today. They haven't changed very much. They still work. Okay, so what we're all here to talk about today is how has vaccine technology matured? How has it changed? What's new since 1955, right? And the answer is a lot. There's been many, but there's 3 major new vaccine technologies that make use of modern practice in biology, molecular biology, and biotechnology that weren't possible 100 years ago, even earlier. And so these 3 different technologies are called recombinant protein vaccines. A good example is hepatitis B. And I'll just briefly go through how it works. So we learned how to express virus proteins in bacteria that we can easily culture in a lab. Okay? So we take the sequence from the hepatitis B targeting protein, which decorates its surface. We clone that into a piece of DNA that we can put into E. coli, which we can culture very easily in a lab. And then we purify that hepatitis B protein. Now, you're never going to get hepatitis B from just one of its proteins. It's not a whole particle. But if you have that purified protein and you inject it into somebody, it'll mount an immune system response against that protein. And that could be strong enough to give you long-term protection. And for hepatitis B, it is. Now, whether that works or not depends on the properties of the protein you're trying to produce. It takes a long time to make some of these vaccines, but this you can make on the order of a couple of years. The next vaccine technology, which some of you may have actually had, is the viral vector technology. So if you had the J&J vaccine, or the Janssen vaccine for COVID, you had a viral vector vaccine. So what happens here is we take a virus that doesn't cause strong disease, usually adenovirus or adeno-associated virus, And we engineer that virus to express that coat protein, that targeting protein, from the virus that we want to inactivate. In the case of COVID, it's the spike protein. In case of hepatitis B virus, it's this protein right here. And so then we culture that virus in cells, human cells, or monkey cells, depending. Purify that virus, and we inject it into you. And then your immune system is activated, but you don't get disease because you only have one COVID protein, that spike protein, and you have no other proteins from the coronavirus. And this virus here, the adeno-associated virus, doesn't cause disease. On its own, you might not even mount an immune response against it. Okay? These also take a long time to engineer and produce. And now the newest technology that I'll talk about, and what I'll spend probably the rest of our time talking about, are the mRNA vaccines. So who knows what mRNA is? Who's heard the word mRNA before? Most everybody. Okay. When I started my career, this is a funny story. When I started my career, I went to a meeting of the RNA Society. And this is a scientific professional society that meets once a year to talk about all different flavors of RNA research. And I was wearing a name tag. It was at the University of Wisconsin-Madison. I don't know if you've ever been there, but they have this beautiful patio outside of their theater, outside of the Memorial Union building. And it overlooks Lake Mendota. And it was a beautiful sunny summer afternoon, and I needed a break from all the dark rooms and the talks and the hundreds to thousands of people. So I went outside with a friend, and I'm sitting on a table on this patio wearing my RNA Society badge. And a couple walks up to us and asks us what RNA stands for, right? And then he asked me, is it Registered Nurses Association? I said, no. RNA stands for ribonucleic acid. We're scientists. We do experiments on RNA. We're here for our conference. And then he said he did experimentation with acid too, but it didn't make him a scientist. I said, that's fair, right? Now when I come to a talk like this and I say, have you guys heard of RNA before? Everybody knows what I'm talking about, right? It's become part of the public lexicon. You hear news stories about RNA viruses and pandemics and RNA vaccines. And what is it? What is it all? What does it do? So mRNA is something that our bodies produce. It's the way that we decode the genes that are stored in our DNA. Stop me if this gets a little bit crazy. What's in these mRNA vaccines is RNA that we transcribe in vitro in a laboratory. What that means is no cells, no animals, no monkey livers, no sheep brains, nothing. No bacteria, nothing. Just nucleotides, an enzyme, and a great big vat. That's how these guys get made. They're then encapsulated with a bunch of different lipids, just like the lipids that are in our membrane. And what these lipids do is they attach themselves to cell surfaces the same way that viruses do with those targeting proteins. And the lipids merge into our cells and release the RNA inside. And so what these lipid nanoparticles do is they protect the RNA from enzymes that are all through our bodies, all in our blood, that would destroy it in seconds. The other thing that these lipid nanoparticles do is they aid entry into the cells nearby where it's injected. Then our cells can engage with the mRNA the same way that our own mRNA engages, and we produce a viral protein that gets presented on the surface the same way a virus would have a protein presented on the surface. And that activates our immune system. Okay. So all 5 of the technologies that I've talked about are still in use today. Attenuated live virus vaccines, inactivated killed virus vaccines, recombinant protein vaccines, mRNA vaccines and viral vectors. So yellow fever, smallpox, measles, mumps, rubella, chickenpox, rotavirus, those are all live virus vaccines. If you're like me and and you're immunocompromised, you have a disease and take a medication that limits your immune system, you have to be a little bit careful with those guys. Most of the influenza viruses, the influenza vaccines that get dosed are heat-killed and activated. Recombinant protein vaccines, hepatitis B, human papillomavirus, shingles, whooping cough. mRNA vaccines are approved for COVID and RSV. And viral vector vaccines, there's the COVID one, and then Ebola and Zika virus. You remember those? Okay. So let's talk about vaccine risks, right? So I just mentioned one. I take a medication called Remicade, which weakens my immune system. I do this because I have a disease called ankylosing spondylitis. And so I take this medication, my immune system is super active, causes all kinds of inflammation, pain up and down my spine, palms of my hands, rib cage, heels of my feet. I take this medication, it turns down my immune system, and I feel great. Now I can stand up here and give presentations, no problem. I take this medication every 6 weeks by IV infusion. But I'm immunosuppressed, which means if you inject a live virus in me, even if it's a weak one, well, I should, make sure my doctor's okay with that. So that's what I do. Some vaccines can cause infectious disease in the right people like me. Sometimes you're allergic to something that's in the vaccine. So most influenza vaccines are made in eggs. Some people have egg allergies. Some people with egg allergies respond to the stuff that's in this vaccine. You don't want that. That causes problems. And so that's why there's multiple kinds of influenza vaccines. Sometimes you have what's called reactive arthritis. Very, 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 very rarely, somebody gets a flu vaccine. They'll develop an autoimmune response. They respond to that vaccine, but the antibodies that they make also start attacking some of their own proteins. That's called a reactive phenomenon. And a very good example of that is Guillain-Barré disease, which is a very rare but real disease. And sometimes you get severe inflammation. Very uncommon, depends totally on the vaccine, but it does happen, right? So I want to, set the record straight. There are risks of injury from taking a vaccine. More often than not, the risks of being infected by the virus far outweigh the risks of taking the vaccine. But it is something to be aware of.
There are risks of getting in your car and biting you.
Oh, yeah. As we talked about at the rally last week. To put the risks in perspective, as I said, I'll get you in just a second. Your odds of dying in a car crash are 1 in 100. Your odds of getting myocarditis from a COVID mRNA vaccine are 1 in 10,000. So just so you can tune your risk tolerance. Yes.
Well, I'm talking about reactions. Many people have a bad reaction, particularly to the shingles vaccine. Can you please explain why?
Let's see if I can do that. So shingles, I think, is a recombinant protein vaccine. The virus that causes shingles is also very similar, if not identical, to the virus that causes chickenpox. So most of us have had chickenpox or have been vaccinated for chickenpox, except for Brandon. But I've had chickenpox. And so that means living inside my immune system is a memory of that infection. And so when we vaccinate with shingles, we're vaccinating with proteins that we have this memory for already. And it activates a stronger immune response than other vaccines do that we're completely what's called naive to. We've never experienced it. That's one reason. Another reason is for vaccines to work well, especially these recombinant protein ones, the protein has to be conjugated to what's called an adjuvant, which is something that your immune system hates. Right? It makes your immune system strong so that it can attack this protein. And for some people, that causes a stronger immune response, and thus that feeling of being sick and icky for a few days. So I think those two things together are why that vaccine is particularly nasty in terms of its side effects. But my immunologist friends tell me you should be happy if you feel sick after you get a vaccine, because that means it's working.
Sean?
Yeah.
Would you mind just staying here where they're putting stuff the rankings?
Right. YouTube. Sorry about that. I'm a wanderer. I do this when I teach lectures over at the university. I like to walk around. Any more questions? Yeah.
One of the things I remember reading about, I don't think it was Shingrix, but I think it was the predecessor of Shingrix. One of the risks with it was it could cause immune amnesia. In other words, it could make your B cells forget.
That's something yeah, I've not heard of that, but then again, I haven't looked into it too deeply. But that would be interesting. I don't quite know how that would work.
It was unusual.
Mm-hmm.
I don't want to be super, but it was something I read in the journal.
Yep. Yeah, I'll check that one out. That's a surprise to me. There's one of the beautiful things about science is there's surprises around every corner. Things that I never expected are now commonplace. So I'll go look into that. That's interesting. Another funny, quick aside story is I gave a speech about vaccines at the rally in Worcester last week, just a short one. And when I got off the stage, a person walked up to me afterwards and told me I did a great job. And I had to blink twice because I recognized him, but I didn't know who it was. Turns out that was my primary care physician. It was like I didn't recognize him without the white coat, and the stethoscope. You know what I mean? And I had to tell him, don't tell these guys I haven't had my shingles vaccine yet, my fall influenza, or my fall COVID vaccine yet. You got the receipts. Don't tell these guys. The truth is I just had one of these infusions. And so when I get vaccinated, I have to time it for the exact midpoint in between my infusions. So 2 weeks from now, I'll be getting my shingles, fall flu, and the new COVID variant vaccines. But I haven't had the shingles vaccine, so I don't know. My brother had shingles, and it was awful. He hated it. So for what that's worth. Okay. So yeah. If I get the flu vaccine prior to the infusion, the flu vaccine takes like a couple weeks for all this whole immune response to develop. And so if I have it right before, and then I get the infusion that weakens my immune system, I won't generate the strong protection against influenza that I would otherwise. Same thing right after. So it's timing it to the point where I'm at my least likely to have a dose of this medication, so halfway in between. I'd also just had knee surgery, and so anytime I have like an operation or anything like that, I have to not take this medication because the risk of infection is higher. And so I'm not supposed to suppress my immune system if my infection risk is higher because they've been scraping around inside my knee joint. So just I have to pay a little extra attention sometimes. Okay, so this is an mRNA vaccine diagram. And basically it has all of the things I've told you about. It has RNA molecules inside. It has an ionizable lipid, something that might be similar to what you find in shampoo, for example. It has these phospholipids like our membranes, and it has these pegylated lipids which stabilize it. And the little black dots are cholesterol, which make it form little droplets of a certain size. And so when I say lipid nanoparticle, what I really mean is tiny little soap droplet. Okay? It's not a machine. It's not a microchip. None of any of those things. It's just a little soap droplet about uniform in size where lipids encapsulate a little bit of RNA to protect them and help them get into cells. Okay. So yeah, what is mRNA? What is this mRNA that we're talking about? So all of our cells have DNA. You've all heard of DNA? DNA, we have, lots of it, miles of it. We have hundreds of thousands of genes. That's probably an exaggeration. Maybe about 100,000 genes in our genome. We have 46 chromosomes, half from mom, half from dad. And every single one of our cells, for the most part, has that full complement of DNA. Okay? The reason why a blood cell is different from a brain cell is different from a liver cell is that which parts of that DNA are active making mRNA are different in different types of cells. Okay? So our brain cells produce the proteins, the mRNAs that we need to transmit signals from one part of our brain to the other. So when I get smacked in the fingers, I don't feel it in the toes. You know what I mean? Wouldn't do for the mRNA that encodes hemoglobin to be floating around inside my brain cells, because then it would be stealing oxygen from my blood. So we have to have the right DNA and the right RNA in the right cells, because all of the cells have the same DNA. And so the process of selecting and producing RNA from a subset of our genes is called transcription. Okay? This RNA, in reality, mRNA, messenger RNA, is the code that we use to make proteins. The proteins are, for the most part, the business molecules of the cell, right? Proteins are what bind to oxygen and transport it. Proteins are what allow cells to stick together. Proteins are enzymes that do chemical reactions. So the act of making protein out of RNA is called translation. So what am I talking about? There's a code in our DNA. Okay? And the DNA just doesn't, there's only 4 bases in it. It's only got 4 letters. Okay? A, C, G, and T in our DNA. When it's transcribed into RNA, it's A, C, G, and then U. Just don't even worry about the difference. It's just subtly different. But it's not the order of those nucleotides, those bases, those letters matters, right? Just like the order of letters in a word matters. And so the words in your RNA and DNA are 3 letters long. And we know this because of experiments that were done in the 1960s by guys like Jim Watson, Francis Crick, and Gamow. Many of these experiments were done right in Massachusetts, down on Cape Cod at the marine biology labs in Woodsville. And basically, the code was figured out, right? There's 20 amino acids that we make. Our proteins only have 20 different letters. But we have 3-letter codes, which means there's 64 possible codes. We know what each one of those possible codes stands for. And so each of these big letters is an amino acid that's coded for by these RNA sequences. We have one that says start. That's this one. It encodes for methionine. We have 3 that say stop making your protein here. And the rest code for amino acids. Now there's 64 combinations and only 20 amino acids, which means some words mean the same thing, right? Just like in the English language. Some words essentially mean the same thing. So if AUG here in our little chart stands for M and start, this is where our code begins. And this particular coding sequence encodes the sequence, my name is Sean. Okay? So if there was a mutation in the DNA, it would change the RNA that gets produced. And depending on what that mutation is, it could change the meaning, right? So if the code right here, UCG, which codes for S, was transformed to AUG in this mutation, the code would read, my name is Mean, which has a totally different meaning, right? And if you get rid of a nucleotide, now you shift the frame. And then it would say, my name is blah, blah, blah. Get it? Okay. So we can write the code. We know what the code is. That means if we can get mRNA into cells, we can program it to make whatever protein we want, right? So if we want it to make virus protein, all we got to do is figure out how to get it in. Okay. This is how mRNA, which is this black line here, makes protein. There's this huge macromolecular machine called the ribosome, which uses an adapter molecule called a tRNA to connect that 3-nucleotide code down here on the mRNA to a specific amino acid. 3 of these transit through the ribosome all at the same time. New peptide bonds get formed, and then it's like a conveyor, just moving right down that mRNA sequence, adding additional amino acids. And the key is this complex, the ribosome, and this little adapter molecule. Okay, how do I know this? Right? We can actually look at it. We have microscopes that are powerful enough to see what these macromolecular assemblies look like in 3 dimensions. Okay, so this is an electron density map of a subunit of the ribosome with 3 tRNAs attached. Now I've just diagrammed in the protein and the mRNA, but we have structures with those guys in there too. They're more complicated to look at, but we can see it with our eye. We can infer it from experiments, but we also have the tools to look at it. It's amazing. And one of my colleagues at the university does this type of research. Okay. So going back to how vaccines work to activate the immune system, basically what an mRNA vaccine does is it skips this part, right? It skips the part where the macrophage or whatever cell engulfs it. We trick cells into taking up mRNA that we give it. They then express that protein the way a viral genome would, and then that gets expressed on the surface, and that activates the immune response. So we've bypassed the infectious disease part. So you can't get sick from an mRNA vaccine in terms of infectious disease. There's nothing in there that can cause an infection. Make sense? Okay, good. Can vaccines themselves, can you develop an antibody response against these mRNA vaccines, right? We don't want that. We don't want antibodies to bind to these lipid particles or to attack the mRNA that's inside of them. Especially if they attack the mRNA inside of them, that would be bad because then you might attack your own mRNAs with antibodies. So the question is, are mRNAs immunogenic? And the short answer is yes. Okay? So I told you last week that the first paper published describing mRNA vaccine technology was published in 1990. Right? This is thought of as new technology. It's over 30 years old. But we didn't get a vaccine in use with this technology until 2020 in the COVID pandemic. 30 years of research and development. One of the things that was being studied is, why is RNA so immunogenic? And it turns out, when I told you that RNA only has 4 letters, I wasn't telling you the truth. Turns out that our mRNAs, one of the things that our cells in our bodies use to recognize our own stuff, as opposed to virus-encoded stuff, our mRNAs have modifications in them. Some of the nucleotides are changed a little bit. A methyl group here, a rotation of a bond there. And those changes help protect those molecules from our own immune system so they don't activate. So our bodies have a really great way of saying, that must be a virus RNA. It doesn't have these changes. Our cellular mRNAs have changes. We didn't know that, really, until about 2010. Okay? And the 2023 Nobel Prize in Physiology and Medicine went to Drew Weissman, and Catalin Carrico, both working at the University of Pennsylvania at the time, when they discovered that if they put modified nucleotides into mRNAs and then did the same experiment that was done in 1990, it no longer activated the immune response. And that enabled vaccine technology, mRNA vaccine technology, because it brought the immune response to these vaccines down to a safe threshold. It's now even safer. We know how to dose with even less mRNA and get a stronger response. And so I won't, go through the chemistry, but the changes are pretty minor. Okay, any questions about that? That's really important. So we had to figure out how to make what's called exogenous mRNA, the stuff that we make in a lab, look more like the stuff that's made in our own cells. Once we figured that out, it's off to the races. Okay. I'd like to switch a little bit into how vaccines get made. Okay, is everybody good with that? Good. So I'm going to compare influenza vaccine, which we take annually, to the COVID-19 vaccines, which we take annually or twice a year. So most of the flu vaccine that you take, and I told you this earlier, is made in a chicken egg. Okay? And just like COVID, there's hundreds, tens to hundreds of circulating strains in the world population every year. In order to make the flu vaccine, the scientists at the World Health Organization, and formerly with the collaboration of the CDC, would pick 2, 3, or 4 of those strains to be their best guess for the ones that would dominate the infectious cycle across the world. Sometimes they get it right, sometimes they miss the boat. That's why the flu vaccine seems to work great some years, but not others. It takes months and months for farmers to grow enough chicken eggs, to be fertilized, to inoculate with enough virus, to grow enough live virus to kill, in order to produce all of the vaccine doses that we use in the United States. Months. And you're limited by that time to just a few different flavors of the virus. You're limited by the expense as to how many different flavors you can make in a year. Okay? I don't know about you guys, but not too long ago, chicken eggs were pretty damn expensive. And, this is one of the reasons why. If something awful like avian influenza wipes out some of these flu vaccine farms, we've made this huge investment for very little vaccine, and the doses become limiting. Okay. So in order to make flu vaccine, we need farms full of chickens making lots of eggs, and we need months of time, and we need to gamble on which viruses are going to be the most abundant. COVID is an mRNA vaccine. It's entirely chemical. No animals are involved in its production. No cells are involved in its production. No proteins. To design the sequence that you want to make, you go to a computer, you type it in, right? You can literally sequence the spike gene from all of the viruses that are circulating. That's pretty much done every time somebody is detected that gets hospitalized with COVID. They end up getting their COVID strain sequenced. You have all these various COVID spike protein genes. You can program the one you want to make a vaccine for into the computer. The computer directs a robot to synthesize a DNA template. That DNA template gets put into a giant beaker, maybe the size of this room, maybe a little bit smaller, along with an enzyme that reads the DNA and makes RNA out of it. Okay? That happens over the course of a few hours. That RNA is then purified away from the DNA, the enzyme, and the unreacted nucleotides and all of the salts that were used to stimulate that reaction. And then they're formulated with these lipids to make these particles. These particles are sized, so they're not so big they clog the needles, not so big that they clog your arteries or veins, to the right size, through filtration and further refinement, and then they're dispensed into vials. So you can make a COVID vaccine in a laboratory in a matter of hours for almost no money. To make doses for humans to be used in patients, you must use what's called good clinical manufacturing processes. And that means a lot more oversight, a lot more protections, a lot more validation, a lot more checks. And that's why it's more expensive to make a dose that goes into a person than a dose that I might use for research. Okay? But there's no reason why you couldn't do this for 10 different strains at once. And in fact, the companies like Moderna and Pfizer who make these things have started making bivalent and trivalent, meaning multiple viruses, in the same bottle. To tell you the truth, there's no reason why you couldn't do this for influenza, why you couldn't do this for yellow fever, why you couldn't do this for polio, why you couldn't do this for measles, why you couldn't do this for smallpox, other than many of those clinical trials haven't been done yet or are in progress. So my hope is to keep this talk neutral, but this, I want you to get the feel that this is a very promising, very robust technology. It's being invested in heavily all across the globe. China has giant plants manufacturing RNA vaccines now. And some of our companies, I talked to the CEO of a company that makes an RNA vaccine that you can dose at 1/100th the amount of the current vaccines. He can't get anybody in the US to touch it because of the current federal climate. So he's been to Brazil, he's been to Vietnam, he's been to China, he's been to Europe, looking for facilities that will make it so that they can do clinical trials. Hundreds of millions of dollars in federal investment that for ongoing contracts to do some of the experiments I just talked about, make mRNA vaccines against other diseases, were abruptly canceled a few months ago. And interestingly, some of those same contracts that were pulled from Health and Human Services were picked up a few weeks later by the Department of Defense, because the Department of Defense realizes that vaccinating soldiers is part of our national biodefense preparedness program. And so that tells you a little bit about who thinks what about the different technologies. Let's see. So 5 different technologies. Some have advantages, some have risks. The advantages and risks of the different technologies need to be considered. Attenuated live virus vaccines tend to generate really strong immune responses that give us lifelong memory and balanced response, okay? But it takes 10 plus years to attenuate a virus so that it works really good, so that it doesn't cause disease, right? And it doesn't work for like upper respiratory viruses like flu or COVID or rhinoviruses that cause colds. And people like me have to be wary of infectious disease. These inactivated viruses, you can get a strong balanced response, but again, it takes a long time and it doesn't always work. Didn't work for yellow fever, might not work for some of these others. Recombinant protein vaccines, yeah, we can make them, but it doesn't always work. So it's a gamble every time. And I've lost my train of thought. Months to years to develop new ones, right? So we had the mRNA vaccines for COVID about 1.5 years before the first protein vaccines were released in the United States. So that's how much more time it took to develop them, to just manufacture them. mRNA vaccines we talked about. The viral vaccines, viral vector vaccines have a lot of promise, but as we learned from the COVID pandemic, as we learned from the COVID pandemic, they had real risks, right? So the British Chad Ox1 vaccine had a very prominent side effect with blood clots, thrombocytoses. And it was so bad that it had to be pulled from the market. And this was something that was not expected. And that's why we do clinical trials. Clinical trials with volunteers. Now, how do you think we do clinical trials for a disease like COVID. Right? In the old days, we would take a prisoner, we'd inject them with our vaccine, we'd give them a month to recover, and we'd fire up some smallpox, and we'd say, "Did you die?" We don't do that anymore. So we need other ways to determine whether or not the vaccine works. Because injecting somebody with a human pathogen is illegal and immoral, right? And so we need to find some other way. And so the metric, yeah. That's one way, but antibodies can be produced that don't actually neutralize the virus. So it's not the gold standard. I hate to use that word because it's been usurped, but it's not the gold standard. What we do is we monitor a large cohort of people for whether or not they get sick, whether or not they get hospitalized, and whether or not they die over a year or two following the treatment. Okay? People are going to get exposed to COVID during a COVID-19 pandemic. And so we look at the rate of infection in vaccinated people versus the rate of infection in unvaccinated people. We look for the rate of hospitalizations in vaccinated people versus unvaccinated people. And we look for the rate of mortality, right? And so by these metrics, using hospitalization and death as criteria, the 2 COVID vaccines that hit the market were 95% effective. By the time the Delta and Omicron wave came around, they were more like 60 to 70% effective. That's because the virus was changing. The spike protein was mutating, and the vaccine was giving us immunity to a virus that no longer really existed. Okay? That's why we have to keep taking boosters. These viruses change quick. Flu is faster than COVID, but COVID does change. Yes?
Back in 2021, what was your response to people who were like, this happened too quick, how can we trust this vaccine?
My response back in 2021 was to tell them I was going to get the vaccine as soon as it became available, that I thought it was the best possible chance that we had to prevent the virus moving from a pandemic virus that we had to deal with for a fixed amount of time to an endemic virus, which is going to be with us for the rest of human history until we find a better way to eliminate it. There are real concerns, right? So I don't want to mitigate that. These vaccines had not been used in people for long periods of time. There were clinical trials that were in the works from before COVID ever came up for other viruses, right? So it's not like it was the very first time the vaccine had made it into humans, but the long-term effects of being vaccinated hadn't been studied because they hadn't been, these clinical trials had just started like 2016, 17, 18. But I told them that the risks of the COVID infection, certainly in 2021, were much higher than the risks of being vaccinated. And then I would cite the papers that were published by the Lancet and the New England Journal of Medicine concerning how well they worked and what the adverse event rate was. And I still say the same thing. Any more questions before I move on? Yeah.
Sean, mRNA has shown tremendous promise with some types of cancer and other diseases. Could you explain that to us and what those might be?
Yeah, so mRNA vaccines use these mRNAs that I told you about to produce virus proteins. And they project these virus proteins onto their cell surfaces, and that instigates an immune response against a virus protein, causing immunity. So how does that work for cancer, right? So when we have cancer, we get mutations in our DNA. And our DNA gets chopped up and rearranged, and depending on the type of cancer and the way it progresses, these mutations can add up and add up and add up. And you end up making what are called cancer antigens, which means changes in your protein content that your cells make that are different enough from your own cells, normal DNA, that your immune system might respond. Okay? Now you can use mRNAs, we can program them to code for anything. So we program them to code for these mutants that are present in your cancer. And then our cells, like our muscle cells, will make that mRNA. They'll make the protein from that mRNA. They'll end up living on that cell surface. And then that activates our immune system to go find cells that have those proteins in them. The only cells in our body that have those proteins in them are cancer cells. And so there's a clinical trial that's ongoing for pancreatic cancer. And I don't know if you guys know about pancreatic cancer, but it's pretty much a death sentence with about a year-ish of from the point of diagnosis to when patients tend to pass. The clinical trial, basically what they do is they sequence the DNA of the patients. They make specific mRNAs that are unique to the mutations that are found in those individual patients. You can do this because it's easy to make these vaccines. And then you treat with those vaccines against the cancer cells that are unique to those patients. And this was a phase 1 clinical trial that's ongoing. It's moved on to phase 2, enrolling more participants. But after 5 years, what was it? 75% or 60%? I think it was 60% of those patients were still alive, which is amazing for pancreatic cancer. Similar results were released for glioblastoma, and there's ongoing clinical trials for many others. There was a really strange result that was published like 3 days ago that I don't understand, and I have to read more into. And that is people that were vaccinated with COVID, mRNA vaccines, and were receiving immunotherapy for cancer at the same time, had about a 60% reduction in the number of cancer cells compared to people that weren't vaccinated while they were undergoing immunotherapy. That was just published in Nature like Wednesday, and I haven't looked into it in enough detail. It boggles my mind. I have no idea how that works or why that would work. I guess the idea is that there's a hidden side of the immune system that we don't know about yet that somehow gets activated. You can read about the labs at the University of Florida that were doing the glioblastoma experiments. A lot of their funding has been cut or canceled. We have labs at the medical school that do mRNA vaccine research that are having a hard time getting grants. I told you about the $500 million of contracts to do clinical trials for these new mRNA vaccines against viruses. So all this work it's expensive. Anytime you're developing a new therapeutic, it's expensive. The pharmaceutical companies are doing the research. They're making the new products. They're doing all the preclinical testing. And they need to find a government partner somewhere.
And what's the reason why they're saying that they're cutting their funding?
Well, that takes me to the last bit of my talk, okay? And that is, I want to take some time to dispel some of the myths that have begun to emerge surrounding mRNA vaccine technology. And one of the ones we've already talked about is that the vaccines were released without testing, okay? A lot of people said that in 2021, even people that I interact with on a daily basis. My own brother, I don't think, has ever been vaccinated with a COVID vaccine. It's just who he is. But it's not true. Like I said, the first paper describing this technology came out in 1990. And there was 3 decades of preclinical testing in cells and animal models to improve the technology to the point where clinical trials could begin, which was around 2015, 16, 17. Okay? The Nobel Prize Committee selected this technology for the Nobel Prize in 2023 after it was obvious how impactful it was during the COVID pandemic. But all of the vaccines that were used in the United States underwent phase 1, 2, and 3 clinical tests, just the way any medication was. The major difference was that instead of being phase 1 and then phase 2 and then phase 3, is that all 3 trials were run simultaneously. And that doesn't usually happen. And so phase 1 is for safety, phase 2 is to establish dose, phase 3 is for clinical efficacy. Well, that was all run at the same time. So they had different trials evaluating the metrics based upon each of those. It was double-blind, placebo-controlled, which means the clinicians doing the vaccination didn't know if they were giving a dose of sugar or a dose of COVID vaccine. All of the data was blinded. Those who evaluated the studies were not the same people as those who performed the study. And that's how you minimize bias. All of those results are independently evaluated by the Food and Drug Administration, and then they make a recommendation as to whether or not it's safe and it's effective. And in both the BioNTech Pfizer vaccine and Moderna mRNA vaccine, the FDA decided to go with an emergency use authorization until longer-term studies could come in for long-term efficacy, reinfection rates, that kind of thing. So people could get the vaccine, but it didn't receive official approval for like another year. That's the history of that. Okay, so they were tested. Some of the roadblocks to rapid testing were removed, but all of the same safety safeguards were there. All the same clinical endpoints that are used for evaluation were exactly the same. Okay. This is my least favorite one. I started seeing this phrase that the mRNA vaccines are dangerous and have killed more people than COVID online about March-ish. And I have a feeling that this is part of, this has come from a organized campaign funded by Project 2025 to discredit the mRNA vaccines. And it's complete, I mean, I don't know what to say about it other than it's a lie, right? It's just not true. So we talked about lightning. We talked about getting into a car wreck. We talked about the rate of the most common adverse event, which is myocarditis. And what I didn't tell you, but I told you last week, is that having COVID gives you a higher risk of myocarditis than getting vaccinated does. And that's true. And the estimate for how many lives these mRNA vaccines saved between 2021, when they were first released, and 2022, end of year, is about 3.2 million. And that's just in the United States. They were used globally. I think the reason why this has come up is that there exists this thing called, what's it called? VAERS, V-A-E-R-S. It's self-reporting of adverse events after you've been vaccinated. So we well, not we, but medical professionals encourage their patients, if they experience something unusual, to contact this system and report it. And so you have a new vaccine against a new disease that was given to some people against their will because they needed to get it in order to go back to work, vaccine mandates by employers, et cetera. That led to a spike in reporting of adverse events. Now, in order to be real, it has to be clinically confirmed. So if somebody gets pancreatic cancer and had a vaccine 2 weeks before they got pancreatic cancer, they might be inclined to think that they got pancreatic cancer because they were vaccinated. But that's probably not true. And in order to evaluate cause and effect, you have to look at the rate of pancreatic cancer incidence in the vaccinated population versus the unvaccinated population versus historical rates. And then you can make correct evaluations. So the people who think the vaccines are dangerous are looking at the raw numbers in this adverse event self-reporting database, but they aren't doing the correct normalizations and statistical comparisons to adjust for what essentially amounts to spurious reporting. Okay? These reports are taken seriously and investigated, but they're not always what you think they are. Yes?
I read through some of the report of the virus thing, and I ran across two. The report was made them feel like the Hulk. So, you know, you gotta take these things with a big grain of salt.
we're all human and we're all prone to thinking, trying to do, we are deductive reasoning beings. We walk around, you know, like I took this geometry class when I was in high school. And the teacher wanted to educate us about deductive reasoning. And this was in the late 80s. So he walked up to the first row of students in the classroom and he smacked the first kid with a ruler. And then he walked to the next kid, smacked that kid with a ruler. And then he walked to the next kid, raised his arm, and the kid went, no, 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 no. And he said, that's deductive reasoning, right? So we are built to do deductive reasoning in order to avoid stressful, dangerous situations. But it's not flawless, right? We draw cause and effect out of correlation all the time. One of my favorite spurious correlations is the number of pirates globally has decreased as a function of time from the 1600s to now, and the average global temperature has increased over time from the 1600s to now, which clearly means that the increase in temperature is changing the amount of piracy we have in the world. So there's databases, there's like great websites of all these fun, spurious correlations. So we have to be very, very careful. And in order to draw meaningful conclusions out of databases like this, you have to have the right training. I don't have it. But there are statistical experts that do. And so I listen to them. Okay. Myth number 3 is that the viruses contain microchips that the government can use to track you using, 5G? 5G tower. They do not. The viruses, the vaccines, the ingredient list, like any other medication you take, is written right in the packaging information. They're soap droplets with mRNA in them. We do. We definitely do. We each have a little supercomputer in our pockets, which is kind of amazing. I watch a lot of car repair videos on TikTok with mine, which may be a little embarrassing, but it's the truth. So I think this comes from a misunderstanding of the term nanoparticle. Some scientist somewhere thought it would be way cooler to use nanoparticle in their research paper than soap droplet. And because of that, this myth persists. And that's too bad. Vaccines alter your DNA. They don't. They can't. It's pretty much impossible. Your DNA is held inside a compartment in your cells called the nucleus. For the vaccine mRNA to get into your DNA, it has to transit the cell membrane. The mRNA has to make it into the nucleus, which it doesn't do. And then that mRNA would have to be reverse transcribed from RNA back into DNA form. Your DNA would have to be cut, and then this little reverse transcribed piece of DNA would have to integrate. Which is exactly how HIV infects you, by the way. They program the enzymes necessary to do this. We don't encode them, but some viruses do. I think where this confusion comes from is there's another technology that's come out at about the same time called CRISPR. Has anybody heard of CRISPR? Oh, yeah. Yeah. I love CRISPR. I use CRISPR in my lab all the time. It is a tool that scientists can use to change the DNA in your cells. Okay? It is literally designed to do the things that people are worried that the vaccine is doing. And it includes an RNA component. It's a specialized small piece of RNA that targets this enzyme to whichever gene you're trying to edit. So this is now being used in clinical therapeutics as well. So has anybody heard of beta thalassemia, sickle cell anemia, things like that? So people that have sickle cell anemia have a mutation in a gene called HGB, or beta globin. if you have two copies of the beta globin gene, one from mom, one from dad. If both of those copies are mutated, you can't make beta globin, and your hemoglobin doesn't form, and you have real trouble transporting oxygen about your body, right? That's what beta thalassemia is. If you have a specific mutation, it causes the beta globin gene to make protein that's folded funny. And then that folded funny protein aggregates in your blood vessels, and that causes the vaso-occlusive crises in sickle cell disease. It's a different mutation in the same gene. So it turns out, this is a bit of an aside, but it's cool. Everybody that's born has a gene that's very similar to the beta globin gene on the same piece of DNA right next door called gamma globin, maybe delta globin, I can't remember. Okay? That's fetal globin, right? That's the protein that your body made when you were a fetus that let you steal oxygen from mom's blood. Okay? When you are born, your body shuts off making mRNA from that gene and never turns it back on. And so you're stuck with beta globin. So you could develop completely normally, and then you're born, and you have two bad copies of the beta globin gene, you're screwed. If we could figure out how to turn on fetal globin in these patients, you could cure this disease. And they've done that. There's a specific protein which represses making mRNA from this gamma globin gene. We can break that repressor gene using CRISPR. And now these patients are fine. And so I think it was 2024, even in January, the first clinical report of the long-term effect from using a CRISPR therapeutic. Bottom line, for the purpose of today's talk, CRISPR therapies are coming. They're going to be awesome for people with genetic diseases like Huntington's, sickle cell disease, these less common but really nasty genetic diseases. As long as we can figure out how to get the CRISPR to the right cells and modify enough of the DNA, we can fix it, which is amazing. But it's not what vaccines do. It's a totally different technology. And then the last myth is that the mRNA vaccines stay in your body for years and that they go all over your body. It's simply not true. Anybody want to hazard a guess? Can you see on the screen? Don't look, no peeking. How long the mRNA vaccine persists in your arm? Years? No. Months? Weeks? It's about 24 hours at most. Hours to a day, at most. And these are the heavily modified protected RNAs that don't degrade as fast as the naked mRNAs. You know how long the naked mRNAs, like the 1990 experiment, last? Minutes. Okay? You can detect the presence of these mRNAs in patients that have been injected for about 24 hours. It's highly localized to the muscle that it's injected into. Sometimes you see a little tiny bit of it in the nearest lymph node. That's it. You don't see it in the brain, you don't see it in the liver, you don't see it in tissues it's not supposed to go to. It stays local.
Is that related to the requirements for very cold storage?
No, the reason why these are stored cold is because mRNA has a tendency to fall apart on its own. It's a very unstable molecule. That's why we store our information as DNA instead of RNA. The DNA is just a lot more chemically stable. With a little bit of heat and a little bit of base, an mRNA will fall apart completely in seconds. So it's stored at, the Moderna was formulated in a way where it could be safely stored at -20 for months. Pfizer needed ultra cold storage, -70 for months. But it's just the nature of the beast. It may be possible to find a way to dry it all down and store it longer at room temperature as a powder. But as long as there's water there, maybe a little bit of base, a little bit of heat, your RNA is at risk. Okay. So lest you think the anti-vax cartoon I showed you in the beginning was a one-off, I went and I found a couple books from this guy. He's a medical doctor from the early 1800s. This book that he published is called Cowpox Inoculation, No Security Against Smallpox Infection. And it's a very, very long book. It's in the public domain now. You can download it and read it for yourself. You'll have to figure out that the F's are actually S's in Old English writing. But it's filled with cartoons like this one, which is a little boy who's growing ox-like features on this side of his face. So he calls this the ox look. And here's an anti-vaccinations flyer where vaccines are depicted as a giant snake coming after your baby with a ghost and a skeleton. And here the vaccines are some kind of mermaid monster beast coming to get you. And there's been even whole books on how to avoid vaccination. And so anti-vaccination sentiments are as old as vaccines themselves. And anti-vaccination sentiments have persisted through the current day and age, even when the disease that started vaccine technology has long since been wiped out. And I think it says more about human nature than it does about vaccinations and vaccine technology. So it's easy to be afraid, and it's easy to not trust. But I would encourage you to trust the researchers, trust scientists, medical doctors, for the most part, right? There have been some very big mistakes that have been made along the way. But I think in most cases, we're not out there trying to make a buck. We're out there trying to help people. I know that I am, although if you want to buy my book. Okay, so the take-home messages. Vaccines work. They protect your family. They protect your kids. They protect your neighbors. There's this concept of herd immunity. If enough of us get vaccinated, then the virus has nowhere to go, nowhere to grow, unless they have an animal reservoir. Not all vaccines are the same, just like not all viruses are the same. So there's different technologies. Some generate a really strong immune response. Some viruses generate a really strong immune response. We just have to live with the fact that some vaccines we're going to have to take over and over and over again, unless we find a better way. And I believe me when I say researchers love finding a better way, because then they can patent it. Any medication that you take, there's risk, right? In the notes for the Remicade infusions I get every 6 weeks, is that I'm putting myself at a slightly increased risk of catching a lymphoma. I say catching, developing a lymphoma, right? I don't want cancer of the blood, but I really enjoy walking. And so for me, it's worth it. I believe that the benefits of that medication far outweigh the risks. This was a fun statistic I learned the other day. I'm working on a manuscript with a friend. I'll introduce you to his picture in a moment. And he pointed out to me that those scary cases of myocarditis that everyone's worried about, almost all of them, like 95 to 99% of people that went into the hospital were released in a day or two, having received nothing more than Motrin for their enhanced inflammation. So even if you get some of the worst adverse events from the mRNA vaccines, they're not causing serious disease. Okay, and if anything, please believe that we're not out there injecting you with infectious agents, hoping that you don't get sick after we vaccinate you. We want your informed consent. And by we, I'm talking about the medical doctors. I don't do any of this stuff. I work on small invertebrate model organisms. I care about developmental biology. Clinical trials are designed and vetted and must be approved by the government, set up in a specific way as to produce interpretable data, and it's independently validated. Okay, so we try to mitigate the risk that our intrinsic biases, which we all have, influence how we think about the outcomes of our experiments. And I want you to know that, if nothing else. And I'm a little bit early, but I'm happy to take more questions if you have some. Yes. Oh, well, because do you want an honest answer or do you want what I think is what I think? So the head of Health and Human Services has been a longtime anti-vaxxer. He's also the kind of person who has lived on the fringe side of science for some time. He supports fringe theories. He popularizes them through his social media. He now directs the agency that selects which grants get funded and which don't. And he's made it his business to replace the people in charge who make the decisions on which vaccines get recommended to be given and which vaccines get funded. So I think a lot of it is his private crusade, although part of me believes that the COVID pandemic was awful for us all, right? The COVID pandemic was awful for us all. Scientists, patients, average everyday people. My now ex-wife, she works at Jewish Healthcare on the west side of Worcester. And in the early days of the pandemic, March 2020, she's a dietitian. She was being called in to do patient care because a huge number of their patient population had COVID and most of their staff had acquired COVID. And she was losing patients every single day, right? This was in the very early days of the pandemic. So it was awful. It was absolutely awful. But not everybody experienced the same pandemic, right? So people who lived in New York City saw the worst of it. People that lived in Massachusetts, we saw a pretty bad pandemic too. People that lived in Italy and in England and in China and in Japan experienced a pretty bad pandemic. Other parts of our country, it was slower and it took longer to materialize. And the impacts of the lockdown, being out of work, and the impacts of the mask mandates were real and meaningful and affected people's lives too. And in many cases felt worse than the actual viral pandemic did, right, in that given community. I do a lot of work with people in recovery from addiction. And when the pandemic hit, all of the meetings closed down for people in addiction recovery. Many people relapsed and died, right? And that is a real consequence of policy driven by the pandemic outbreak. And so there's a backlash. And I think it's natural that there's a backlash. I don't think it's a rational backlash that's founded in science, but scientists and the mRNA vaccines in particular are the focal point of that backlash. So I'm just being honest here. And I think by catering to people that are unhappy with the outcomes of the pandemic, politicians are gaining an edge politically. Does that make sense? Right, so you get more people to vote for you if you say you're going to punish the people that did this to us.
What can we do to help what's happening with the defunding of research and what's happening at CDC and NIH with the defunding? Is there anything?
Well, yes. The worst thing happening in science right now is the shutdown. And so I was supposed to review NIH training grants on October 20th and 21st. I was set to chair a study section, which is a review panel, which reads through a bunch of these grants and provides scores, which the NIH then uses to decide which aligns best with their priorities and scored well, right? Those did not happen, and so those grants are sitting there unreviewed. I submitted a grant a week later to my own program to keep my lab going. That has been sitting on some computer version of a desk unlooked at since I submitted it. And so as long as the government remains closed, the NIH is at a total standstill. No new grants will be funded, no grants will be reviewed. And it's particularly bad for us this year because there's only 3 review cycles per calendar year. We missed the first cycle because when the current administration took power, they instituted a new federal branch called DOGE, which came in and shut down all of research funding while they were evaluating it. And that took months, and no new grants were funded. Some grants were terminated. Funds weren't being dispersed on grants that had already been awarded. So we've only had 1 review cycle this year. And what that means is we can't really do our work. There's going to be a huge backlog, and it's going to take maybe a full year to catch back up. So one thing you can do is talk to your local representatives and convince them to get back to work. I don't know how to do that, but it is important for science. Those of you who are of means can donate to nonprofit organizations that fund research. There are many. You may find some that are in a wheelhouse for something that you like. I've donated to the Multiple Myeloma Research Foundation because that's the cancer that killed my father. You can donate to specific departments at the medical school. My department is Biochemistry and Molecular Biotechnology. There's a way to donate directly to that. That's not going to solve the problem, but it may help save a job or two.
Is it true that China is recruiting our scientists as well as other European countries, as well as European countries?
Yes and no. So China is investing very heavily in academic research right now. They're building new universities, they're founding new departments, they're putting out international calls, recruiting scientists. There are many scientists from China who have trained in the United States that have decided to go back. There are some scientists who are American or European that are moving to China to set up their labs over there. So that is happening. Mostly it's Chinese national scientists that have been in the US for a while and had planned on staying that are leaving, but there are some others. Europe, it's not as robust, and the reason is because Europe doesn't fund science as well as the United States does, or did, more accurately. And there's a great call from European funding agencies and political bodies, governments, to use this opportunity to bring European scientists home, or to elevate European scientists to greater stature by providing them with more funding to do their research. So there are some labs that are moving to Europe or Canada, but mostly they're saving their money for their own. There's a lot of people that are shutting down their labs and trying to get jobs in biotech companies or pharmaceutical companies. Those jobs are scarce right now. So I don't really know. It's a tough time to be a scientist. I'm not going to lie. I stay up at night thinking about the future. The good news for me is that I have 2 NIH grants right now, one of which runs out in March, but the other one runs out in 2029. So I'll have at least some lab through 2029, unless that grant gets terminated for some reason. Funding priorities change. Any more questions? Here and then here.
I was just going to say, you talked about R.K. Jr. Earlier this week, Frontline, PBS Frontline, did a 2-hour documentary on him. Very interesting.
Yeah,
It's scary.
I'll check it out, but I disagree with almost everything that he has to say policy-wise. Here and then back there?
I have of a dozen things. I live around here. I live in Massachusetts since 2000.
Mm-hmm. geez.
who leans against your belief.
Mm.
But I'm mainly upset that there's almost no civil dialogue between progressives and I'm conservative, but I'm a critic of the official source. My question is, the reason I came is that you said that but Robert Malone is one of the main people that he replaced him. Now, my understanding, maybe you shouldn't have the right to say this, but my understanding, when you had tens of millions of views on Joe Rogan, is that he is the developer of the mRNA vaccine. That's probably, well, what do you say about Malone and mRNA?
So I'd say that's a bit of an overstatement. He was definitely involved in the early days of mRNA vaccine technology development. When I talk about the first papers that were published in the early 1990s, he was involved in some of those. But the key transformative discovery that turned it, the two key discoveries that turned it from an interesting idea into practice was encapsulation in lipid nanoparticles and the modified nucleotides so that the RNAs looked more like our own. And that's what made it a plausible technology. So I think he overstates it when he says that he's the inventor of mRNA vaccines.
I don't know if he uses that exact term, but it seems that way.
Yeah. You know, everybody's got a right to their opinion. My opinion is based upon the literature that I've read, which includes the clinical trial data and conversations with others and the experiences that I've seen. I try to be as honest and forthright as I can be with what I say. I don't know Dr. Malone personally. I don't know why he says some of the things that he says, but it's not the consensus in the field. Did you have a question back here?
I think a lot of it just was driven what you were saying about the viruses. That they're willfully ignorant. That have they? And then they make you slow it up. Like you were talking about, 5 minutes. No, it's crazy.
I mean, it's hard to know, right? So I like to think of myself as a person who crosses a lot of communities. So like I said at the beginning, I'm a regular everyday guy. I play music, so I intersect with the local music community. I love country music. I love hairband music, although I don't qualify anymore. My dad was an Air Force veteran. He retired after 20 years. And then he was an outlaw biker type. And so I've been to parties with lots of bikers and talked to them. I talk to the music scene people. I have friends that cross a lot of spans. I do a lot of motorcycle repair. I'm one of those guys. I'd say for the most part, people are genuine in their beliefs. Where things seem to fall apart are the sources of information, right? You can describe something in an entirely plausible and convincing way, but it may not be the truth. And until we can come to consensus on what the truth is, we're going to continue to talk past each other instead of to each other. And I don't know how to convince people that some sources of information are more robust and reliable than others. Sometimes I don't know if I should. But I think that's the fundamental problem. I don't think there's any malintent, or maybe there is, but I don't think the average person is filled with malintent. I think there's, we just can't agree on who's telling the truth. Yeah, and then I'll go over here.
Dr. John Campbell is somebody I've watched on YouTube. Every few months he presents excess data. It's from our world in data. He goes, he's very demure, he goes into great detail of why so many countries in the developed world have significant excess deaths after 2021, which is his argument that there are more deaths from the vaccine than there were from COVID. Are you familiar with Dr. John Campbell's work?
I'm not familiar. I'll have to look into his analyses and what he's done.
He's got a PhD in health information. He taught nursing school for decades. He's British, he's very good. And he has a new presentation on excess deaths every 3 months now.
I'll check it out. I will say that the excess death data that I followed in the early days of the pandemic, presented by local scientists, painted a different picture. But that was, again, the early days of the pandemic when I was following the spread across the United States. So I'm not as familiar with what's happening now.
May I correspond with you by the book?
Yeah. Over here.
Oh, let's get back to science a little. So we have these messenger RNA going to the ribosomes to produce proteins that stimulate your immune system.
Yep.
Now, is there any mRNA vaccines that actually produce enough memory that you wouldn't have to get inoculated every year?
It's unclear, right? So the clinical trials that are ongoing are against influenza. There's an approved one against RSV. It's unclear whether, like, if a measles or mumps or rubella vaccine were made out of mRNA, if it would confer the same kind of long-lasting antiviral memory that the current attenuated live virus vaccines do. That's why we got to do the clinical trials. That may be a limitation of the technology. We just won't know until that data comes in.
Since our case community didn't believe in the vaccine of the measles, they actually go down to Texas when a 7-year-old or a 9-year-old died of measles. They're like, gone on since then, or measles death.
So the last I looked up measles was about a week ago before the rally. And the current outbreak totals in the United States sits around 1,600. Many of those cases, I think, are in North or South Carolina. I can't remember which. And about 10% of those have led to hospitalizations. But that's about where it sits now. Measles has a much higher R0 than COVID. And what R0 is it's a statistic that defines how many people the average person will spread their infectious disease to. So for COVID, in the early days of the pandemic, it was estimated to be around 2.5. For measles, it's 20. But that's considering a completely unvaccinated, susceptible, naive population. I do not believe that the current measles outbreak has an R0 that would approach anywhere near that number. But that's what's possible in unvaccinated communities. Okay, let's wrap this up. Thank you guys for coming. I'll stick around for a little bit if you're interested in the book. And thank you.